molecules compared to extracellular fluid, and it has diverse molecules. It serves as the site of physiologic cellular processes vital for life. And so all those things we talked about that are responsible for life at the cellular level take place in the cytoplasm. The molecules in the cytoplasmic matrix include inorganic solids such as, so they are not organic and they are dissolved in solution, such as calcium, right? So calcium is dissolved in the cytoplasm and it's inorganic. Calcium, sodium, potassium, chloride, etc., etc., zinc, manganese, magnesium, phosphate, phosphorus. You have intermediate metabolites. And so you're in the process of synthesizing a protein. That protein fragment is sometimes suspended inside the cytoplasm. You have carbohydrates, lipids, and proteins. And so they're coming into the cell. They're being sent from one place to another place to be used. They'll be found inside the cytoplasm. And you have RNAs. RNAs have been made from the DNA, and they are being used to carry amino acids to synthesize proteins, or they are being read so that the instruction facilitates protein synthesis. The solute concentration in the cytoplasmic matrix is tightly controlled, and it influences the rate of metabolic activity. So this is one of the things you're going to learn about in physiology, that within the cell, you keep the concentration of various solutes, intermediate metabolites, and biomolecules within very narrow ranges. And when you exceed that range, the cell becomes disease, right? And so that narrow range is essential for life, and it facilitates the rate of metabolic activity. So you're going to learn about homeostasis in physics. The matrix suspends organelles, the membrane-bound organelles. It suspends the cytoskeleton, and it suspends inclusion bodies. So inclusion bodies are not cytoskeletal elements, and they are not membrane-bound. So things like glycogen exist within the cytoplasm as a large glycogen macromolecule, right? They don't exist in the cytoplasm as a membrane-bound glycogen vesicle. The cytoplasm and nucleus have distinct functions. So they don't do the same thing, but they work together to maintain cellular viability. So if you have cytoplasmic disease, it will affect nuclear viability. And if you have nuclear disease, it will affect cytoplasmic viability. A cell cannot run without these two being healthy. That makes sense, right? That's like really brief. It makes sense. So anyone who sat down and read these notes before today's class, you're just breezing through. Yeah? Which is called the nucleolus. And the nucleolus is the site for ribosome synthesis. A ribosome is a tiny cellular organelle that acts as a molecular printer for proteins. And so you feed a ribosome instructions about the amino acid sequence in the protein, and the ribosome facilitates the stitching together of those amino acids to make a polypeptide. And ribosomes are synthesized inside the nucleolus. And then surrounding the nucleolus is chromatin. And chromatin is simply very densely organized DNA. Right? So that's about it. Each cell in your body except the gamete contains the full amount of DNA that you have. So any of your cells contains all your DNA, right, packed inside it. And the reason some cells are different from others is some regions of DNA are switched off and some regions of DNA are switched on for each cell to exhibit its unique properties. And so for example, the epithelium of the skin and the epithelium inside the mouth are the same type of epithelium. They are a stratified squamous epithelium, flattened cells in layers. But in the skin, the epithelium produces large amounts of keratin, and in the mouth, the epithelium does not produce keratin. That difference is because the keratin region in the epithelium of the skin is switched on, and the keratin region in the epithelium of the mouth is switched off, right? But they both contain a keratin region. And so you have chromatin inside the nucleus, and then surrounding the nucleus is what is called the nuclear membrane. And the nuclear membrane is a bilaminar membrane. So it's based on the same template. It's a phospholipid structure that has two layers, and it separates the nucleus from the rest of the cytoplasm. And within that nuclear membrane, you have regions called nucleoporins. And nucleoporins are channels that lead in and out of the nucleus, and they regulate the movement of material into the nucleus and out of the nucleus. So nothing enters the nucleus unless it crosses through a nucleoporin, and nothing exits the nucleus unless it passes through a nucleoporin. Chromatin inside the nucleus is organized in two ways. It can be dense chromatin. I wish I had an image, I don't. But it can be dense chromatin or loose chromatin. Dense chromatin is called heterochromatin, and loose chromatin is called euchromatin. Euchromatin. And the difference is that the enzymes that read DNA and make copies, so DNA synthase, or the enzymes that read DNA and make RNAs from it, so mRNA synthase, access euchromatin better than they access heterochromatin. And so the regions of a cell whose DNA is being actively expressed, or the regions of DNA that are actively expressed in a cell, exist as euchromatin. The regions that are not being actively expressed exist as heterochromatin. And an important region of heterochromatin in women or in females is the second X. Genetic material is composed of DNA and proteins that organize the DNA into nucleosomes, chromatin, and chromosomes. And so DNA is a nucleotide strand, and DNA is very long. I like to contains 2 meters of DNA, right? Now you have many cells in your body. You have like 100 billion cells or something. You have far more than that. If we take all your DNA and we stick it together end to end, it will go to the moon and come back. Yeah? Now and yet, wewe ndio huyu, sindio? You don't extend to the moon and back. So each cell contains 2 meters of DNA. Now, the largest of your cell nuclei is 20 micrometers long. A micrometer is a millionth of a meter, right? And the cell nucleus is 20 of them. And inside the cell nucleus, you will pack 2 meters of genetic material. The question is, how do you do that? Wrap it around proteins to reduce the length of that molecule. And so there are proteins inside the nucleus called histones. And we wrap DNA around histones, and we make a string that has beads in it, right? And that's called the string on bead appearance of DNA. And so you make the string, so you initially have into a helix, you shorten it a little. And then you take the helix and you wrap it around a histone protein to make what is called a nucleosome. And then you take your nucleosomes and you twist them even more, right? And as you twist them, you form chromatin. And then, at the time of cell division, you can take segments of that chromatin and organize them into chromosomes, right? And so chromosomes only exist at the time of cell division. They show up at the beginning, towards the end of prophase. They exist through metaphase and telophase, and they metaphase and anaphase, and they disappear by the time of telophase, right? And so chromosomes only exist during nuclear division. And so that's how you modify DNA to make sure that your 2 meters of genetic material fits inside this region that is 20 millionths of a meter, right? And that modification is fancy stuff. It's quite impressive to think about it, yeah? Like you wrap it and wrap it and wrap it. How many 20 micrometer units are there in 2 meters? So 2 meters are 2 million micrometers. True? So you divide that by 20 and you get 100,000, right? And so you shrink the DNA 100,000 times to fit it inside the nucleus, and you use that process. At the time of metaphase, the chromosomes exist in duplicate, right? And so one of the things that the cell has done before it enters mitosis is to duplicate the DNA, right? So that what would have been a single chromosome now appears as a duplicated chromosome at the time of cell division. And when you're making the new cell, all you do is you split a copy of the chromosome. But we'll talk about that. Yeah, so this is just to help you understand how chromosomes are constructed. All right, it is lunchtime. Would you guys like to go for lunch?